Hello everyone, welcome back. Today we will continue with phytohormones like cytokinins, ethylene, abscisic acid. So cytokinin is a hormone which is meant for cell division, and ethylene which is important for fruit ripening, and then comes abscisic acid which is commonly known as the stress hormone. So we will discuss it one by one. So let's start. So first, cytokinins. As the name indicate, cyto means cell, kinins means kinases means division. So these are the chemical substances which helps in cell division. So they are basically regulators of cell division and plant growth, and they also prevent leaf senescence. So basically, regulators of cell division and prevent leaf senescence. So this is a class of plant growth regulators or plant growth substances which are active in cell division, promoting cell division, and also involved in various physiological processes like cell growth and cell differentiation. Mainly they are formed in the roots and transported to the shoot through xylem. So they are produced when shoot sends any signal, then they are also formed in the roots, and then they are transported through xylem to the various organs where it is required. There is a chemical named as kinetin, chemically known as 6-furfuryl aminopurine. Basically जो cytokinin है, that is the derivative of a nitrogenous base, जिसका nitrogen basis में हम purines पढ़ते हैं. So it is a derivative of purine, and this is the first known cytokinin and first discovered cytokinin, and it mainly helps in promoting the cell division. And the most commonly found cytokinin is commonly called as zeatin, and it was first isolated from the corn. जो मेज है, मेज प्लांट से ये सबसे पहले आइसोलेट किया गया था, डिस्कवर किया गया था. So this is the most common form of cytokinin. And cytokinin concentrations are highest in the growing young tissues because जो young regions होते हैं, they contain the group of cells which are continuously dividing cells, and they are undifferentiated cells commonly known as meristematic cells. So mostly maximum concentration of cytokinin is found in the young regions, that is meristematic region. So these are some this is the natural occurring cytokinin, that is isopentenyl adenine, and it exists in two forms, that is transzeatin and ciszeatin. Then we have some artificial or synthetic cytokinins like benzyl adenine. It is also known as benzyl aminopurine, and this TDZ. So these two are synthetic cytokinins, basically, which show similar properties as that of the natural cytokinins. So इनका function भी सेम है, जैसे नेचुरल साइटोकिनिन काम करता है वैसे ही ये केमिकल सब्सटेंसेस भी वर्क करते हैं. Next, discovery. So cytokinin like substance was first observed by Haberland in 1913, and he observed that this chemical substance stimulates the cell division. So in 1913, Haberland discovered a chemical compound which stimulates the cell division. Then next, this chemical substance was first isolated by Miller and his co-worker from the herring sperm. Herring is a fish. Herring sperm fish से सबसे पहले isolate किया गया था ये chemical substance. This is the photograph of Haberland, who is commonly known as father of plant tissue culture. So Miller and his co-worker were the first to isolate cytokinin from herring sperm. Herring is basically a fish, and from the sperm of this fish, this chemical substance was isolated, and it was named as kinetin. And the first natural cytokinin was isolated or obtained from unripe maize grains, and this work was done by Latham in 1964, and it is known as zeatin. So this is the most common form of cytokinin. And there are some synthetic auxins, artificial auxins, which show similar properties with the natural auxins. और इसका नाम है benzyl adenine. It is also known as benzyl aminopurine. So this is the artificial or synthetic cytokinin. Next, we have some physiological roles of cytokinin. It plays very important role. As name indicate, it helps in cell division. So first is cell division. It stimulates the cell division in the callus culture. So in the presence of auxin, cytokinin brings about division even in the permanent cells. So already differentiated cells can undergo cell division when there are proper concentrations of auxin and cytokinin. So ये जो दो hormones हैं ये control करते हैं cell division को even in permanent cells. So cell division in callus is unorganized or undifferentiated irregular mass of dividing cells that is mostly used in case of tissue culture. And this callus differentiation requires these both hormones, that is auxin and cytokinin. So this is the picture which shows cell division in case of plant cells. So this cell division is controlled by this chemical substance. So there are various phases of cell division like anaphase, telophase, and finally cytokinesis. So this nuclear division is controlled by auxin, and this cytoplasm division, that is cytokinesis, is controlled by cytokinin. So these both hormones are important for cell division. So basically it induces cell division in the presence of auxin in tobacco pith callus and in pea callus. Callus is an unorganized mass of cell that we use in case of tissue culture. And auxin promote, as I have already told you, it promote nuclear division. Auxin nuclear division में help करता है, while जो cytokinin है that helps in cytokinesis, that is division of cytoplasm. So there is division of liver by these both the hormones, and it involves 3 steps, that is DNA synthesis, then mitosis, and finally cytokinesis. So cell division involves these 3 steps. Next important function or a physiological role of auxin, cytokinin, is cell elongation. So like auxin and gibberellins or gibberellins, cytokinin also cause cell elongation. As we know, these 3 hormones are growth promoting hormones. Cytokinin basically increase the respiratory activities, and greater amount of potassium ions they are accumulated inside the cell, जिससे कि जो osmolytes है और solutes उसकी concentration cell के अंदर increase हो जाती है, that increase the entry of water from the surrounding cell, और जिससे कि cell की elongation होती है, that we have already discussed in case of auxin during the cytokinin induced cell elongation. And it causes expansion in the leafy cotyledons. So जो cotyledons हैं उनकी भी expansion होती है in some plants like mustard, sunflower, and cucumber. So these are this is the function of the cytokinin that helps in cell elongation. So in this picture we can see here, this is the region where the cell elongates because due to the activity of the cytokinin. So this picture shows the various involvement of various types of genes and transcription factors that are involved in this elongation of cell. And the second function is expansion of leafy cotyledons. So these are the leafy cotyledons, first leaf-like structures, and these are known as leafy cotyledons. So their expansion, broadening, is also done with the help of this chemical substance, that is cytokinin. And this picture shows the germination of seeds in case of common bean, that is Phaseolus vulgaris, और इसमें जो cotyledon है वो soil के बाहर आ जाता है. Why it comes out of the soil? Because there is excessive growth of this part, that is hypocotyle, और जब hypocotyle बहुत ज़्यादा grow करता है, it pushes seeds out of the soil surface, and this type of germination is commonly known as apigeal germination. This is known as apigeal germination. While in some plants the seed remains under the soil surface, and that type of germination is known as hypogeal germination. So there are 2 types of germinations. One is apigeal, and this cotyledon comes out of the soil surface, जब ये cotyledon soil के surface से बाहर आ जाएगा. और दूसरा, when it remains in the under the soil surface, that is known as hypogeal germination. Other important physiological role of cytokinin is in morphogenesis, that is beginning of shape. So, cytokinin and auxin's ratio helps in differentiation of callus. Both auxin and cytokinins plays very important role in morphogenesis or differentiation of tissue and organs. In case of tissue culture, when cytokinin concentration is more as compared to auxin, then buds are found, especially shoot buds are found. Shoot buds are found when the concentration of cytokinin is greater than as auxin, while roots are found when their ratio are reversed. It means the auxin, the concentration of auxin is more as compared to cytokinin, then the roots will be found. This was first observed by Skoog and Miller. So high kinetin, that is cytokinin, and low auxin promotes the formation of, promotes shoot formation. Shoot formation होती है जब kinetin की concentration ज़्यादा होती है और auxin की concentration कम होती है. While roots are found when this ratio is reversed. It means जब auxin की concentration ज़्यादा होगी और kinetin की concentration low होगी तो roots बनेंगे tissue culture. So in this picture we can see here, this is the explant, that is a plant part which is used in tissue culture. जब इसको हम nutrient medium में रखते हैं, so it will form the mass of undifferentiated cells, callus कहा जाता है. When this callus is treated with, so in this picture this shows auxin concentration and this is the cytokinin concentration, that is milligram per liter. When the concentration of auxin is high and concentration of kinetin is low, it favors the growth of root system. So roots are formed. When the concentration of auxin is very low and concentration of cytokinin is high, then it promotes the formation of shoot. So this is how the auxin and cytokinin plays important role in callus differentiation in case of tissue culture. Next, differentiation, that is formation of permanent cells from the meristematic cells. So cytokinin induce formation of new leaves, chloroplast in leaves, and lateral root shoots, and formation of adventitious shoot. So these all the events are controlled by cytokinins, and they also bring about lignification, that is deposition of lignin during secondary growth in case of trees, and differentiation of interfascicular cambium. So जो interfascicular cambium बनता है during secondary growth, that is growth in girth. So this interfascicular cambium, that is the cambium जो present होता है दो vascular bundles के बीच में, और ये बनता है at the time of secondary growth. And this interfascicular cambium is also formed by the activity or stimulus generated by the cytokinin. So in this picture we can see here, so this is the one vascular bundle, and the cambium which is present in this vascular bundle is known as intrafascicular cambium, and the cambium which is present in between the two vascular bundles is known as the interfascicular cambium. So this cambium is formed by the activity of cytokinins, and this whole ring of cambium helps in secondary growth, that is growth in girth. So it cuts towards outer side secondary phloem and towards inner side the secondary xylem, and this secondary xylem is commonly known as wood. So most of the dicot shows this secondary growth, that is growth in girth, and it is controlled by the activity of cambium, which is the interfascicular as well as fascicular cambium. Next important function of cytokinin, it helps in prevention of senescence, that is abscission, and this is commonly known as the Richmond-Lang effect. When we put excised leaf in the cytokinin solution, it delays the yellowing of leaves, and that delayed yellowing of leaf is known as the Richmond-Lang effect. So cytokinin helps in delaying senescence. What is senescence? Senescence is programmed aging of leaves, which is associated with loss of chlorophyll. Basically, जो chlorophyll है वो degrade हो जाता है during the process of this senescence, and senescence is the programmed aging of leaves or any other plant parts. It leads to the yellowing of leaves. In winter, we can see the leaves looks yellowish in color due to degradation of this chloroplast or chlorophyll degrade हो जाता है, and it finally leads to the abscission of leaves or fall of leaves. When we put excised, जब हम excised means parent plant से leaf को तोड़ के when we provide it in with cytokinin, जो yellowing of leaves है that can be delayed, and such delay in the yellowing of leaves when we put it in cytokinin solution is known as Richmond-Lang effect. It is the effect named after the discoverer of this phenomenon. And cytokinin delays senescence of leaves and other organs by metabolization or mobilization of nutrients. So basically जो cytokinin है that helps in mobilization of nutrients from the older parts to the younger parts. So this mobilization of nutrients prevents the formation of this abscission zone or senescence is delayed, and it prevents senescence by prevention of degradative enzymes like protease, RNase and DNase. So ये जो enzymes हैं they are not formed in the presence of cytokinin, and that finally leads to the prevention of this senescence. So in this picture we can see here, this is the plant which is without cytokinin. So it shows degradation of this chlorophyll, and it leads to the formation of this yellowish leaves. But when we provide it with cytokinin, it doesn't show yellowing of leaves or degradation of chlorophyll. So cytokinin plays a very important role in mobilization of nutrients that delays the senescence. Next important function of cytokinin is promotion or formation of chloroplast from the etiolated seedling. जो etiolated plants होते हैं, etiolated plants are those plants which are grown in dark conditions. उनमें जो chloroplast formation है that is also initiated by cytokinin. Cytokinin can induce thylakoid formation in the plastids of dark bone seedlings, which are commonly known as etiolated seedlings, suggesting that the chlorophyll development is controlled by the cytokinin, and hence it leads to the formation of chloroplast, which is known as graining process. So this is the type of plastid which is present in dark grown seedlings, that is etiolated seedling, और इसे commonly कहा जाता है etioplast. When cytokinin is present, so this cytokinin helps in formation conversion of this etioplast into this chloroplast, because it helps in development of thylakoids, and this chloroplast can also be converted into chromoplast. It will depend upon the requirement of plant, and gerontoplast, which is formed during aging process, and it is formed by the degradation of this chlorophyll and leads to yellowing of leaves. Next is apical dominance that we have already discussed in case of auxin. So it counters the effect of auxin. Presence of cytokinin in an area causes preferential movement of nutrients toward it. When it is applied to lateral buds, so they helps in their growth despite of the presence of apical bud. So we have discussed this already. जो lateral buds हैं, अगर हम उनमें externally cytokinin apply करते हैं, तो apical bud की presence में भी lateral buds grow करते हैं, and that prevents the apical dominance or effect of auxin. So they thus act as antagonistically against the auxin. So cytokinin works against the auxin, which promotes the apical dominance. जो auxin है वो promote करता है apical dominance को, but when cytokinin concentration is greater, then there is formation of lateral buds even in the presence of apical bud. So in this picture we can see here, this is the tip which is the source of auxin, and it grows lengthwise, and this is known as apical dominance, and while lateral buds are dormant. जो lateral buds हैं वो dormant रहते हैं, means they don't show any growth. But जब हम इसको remove करते हैं, when we remove this tip or apical bud, then it depletes the source of oxygen. जो ऑक्सीजन का सोर्स है वो डिप्लीट हो जाता है. Then there is growth of these lateral buds. But when we apply cytokinin externally in the lateral buds, so they can grow, they can show growth even in the presence of this apical bud. So cytokinin helps in the growth of lateral buds and works antagonistically against the oxygen. Next is seed dormancy. So cytokinin helps in prevention of seed dormancy. Seed dormancy is is a term which is used for the seed which fails to germinate even after providing the suitable conditions for germination. So that condition is known as dormancy or state of inactiveness. Like gibberellins, this cytokinin also helps to overcome seed dormancy of various types, including red light requirement of lettuce and tobacco seeds. जो रेड लाइट की रिक्वायरमेंट होती है टोबैको और लेट्यूस सीड्स की, that can also be replaced by cytokinins. So this helps in germination of seeds. Alone GA cannot overcome the thermos dormancy or inhibitor dormancy, means dormancy caused by high temperature or dormancy caused by some chemical inhibitors which inhibit the seed germination. So addition of cytokinin along with GA opposes overcome this dormancy. So ये दोनों जब present होंगे cytokinin और GA, they can initiate the process of seed germination. So they helps in prevention of seed dormancy. So this is the picture which shows the seed germination, and this type of germination when the cotyledon comes out of the soil is known as apigeal germination. So this is all about cytokinins. Next we will discuss about ethylene or ethylene, which is basically a hormone which is responsible for the ripening of fruit that we can see in this picture. So this is the tomato fruit which is red in color, that is ripened, or this is green in color, unripe fruit. So basically the role of this hormone is in ripening of fruits. So let's start. Ethylene or ethylene, it is a gaseous hormone which is responsible for the ripening of fruits. So it is a gaseous hormone with simplest structure, produced in all plant parts, associated with fruit ripening, and it shows triple response that we will discuss under the heading of physiological responses or physiological roles of ethylene. So this is the structure of ethylene. It was first observed by Gain in 1934 that plants synthesize this gaseous hormone ethylene. In 1935, Crocker proposed that ethylene was a plant hormone and is responsible for fruit ripening. So these are the two scientists which are associated with the discovery and role of ethylene or ethylene. This is the simplest structure of ethylene. The chemical nature of ethylene. As we know, this is the simplest gaseous hormone with a molecular weight of 28 and is well known simplest olefinic gas and has following structure formula that we have already discussed. Ethylene is a flammable, highly volatile, and that rapidly undergo oxidation, and it will form ethylene oxide after oxidation. In many plants, जो ethylene है that can be produced or that can be fully oxidized into carbon dioxide through the ethylene oxide. So this is the simplest structure of ethylene. The physiological roles of ethylene. First, the important role is in fruit ripening. This is the picture which shows this ripening process of banana. This is the unripe fruit and this is the ripened fruit, and during this process the ethylene helps in the ripening of this fruit. In this picture we can see here there are two conditions. One is control condition, that is without ethylene, and these fruits are unripe, while in the presence of ethylene the fruits are ripened. So it aids in, or it imparts in, ripening of climacteric fruits and dehiscence of dry fruits. Climacteric fruits are those fruits which are fleshy and which shows sudden sharp rise in the rate of respiration at the type of ripening. So these fruits are known as climacteric fruits. ऐसे fruits जिनमें rate of respiration बहुत ज़्यादा increase हो जाता है, and they can be ripened even after plucking from the parent plant. They are usually transported in the green or unripe stage, and ethylene is used to induce the artificial ripening of these fruits. For example, in case of apple, mango, banana, these all are the examples of climacteric fruits. And there is one chemical which is named as ethaphone, which is used for the ripening of fruits. This picture shows the examples of climacteric fruits and non-climacteric fruits. So climacteric fruits are those fruits in which there is sudden rise in the rate of respiration during the ripening process. और ये जो fruits हैं वो ripen होते हैं after they have been picked or plucked from their parent plant. और इसमें जो ripening process होता है that is very quick. While in case of non-climacteric fruits, ये जो fruits होते हैं वो तभी mature होंगे, ripen होंगे when they are attached with the parent plant, and the rate of respiration is low as compared to the climacteric fruits. So these are some examples that can be asked in the entrance exams. So we can remember 2 or 3 examples from both climacteric as well as non-climacteric fruits. Next important physiological response of ethylene is it induces flowering in some plants like pineapple, that helps in synchronization in fruit set. So uniformity आती है उसमें flowering के टाइम पे, और वैसे ही synchronized manner में fruits बनते हैं. So it stimulates flowering in pineapple and related plants. So this is the plant pineapple which shows flowering, and it is induced by ethylene. So this helps in synchronizing the fruit set. So uniformity आती है उसमें fruit setting के टाइम पे. So this is the important role of ethylene. Next, it also plays an important role in sex expression. So like oxygen and cytokinins, ethylene has feminizing effect, means it produces female flowers on the sex expression. So it is meant for production of female flowers. The genetically male plants of cannabis, that is marijuana, can be induced to produce female flowers, as we have discussed in case of the oxen. Oxen also do this. Gibberellins, they also form the male flowers in case of cucurbits, while this ethylene produces female flowers in cannabis, that is genetically male plants, in the presence of ethylene. So the number of female flowers and hence the fruit is enhanced in monoecious plants like cucumber. So this in case of cucumber, it also helps in formation of female flowers, and that can enhance the fruit production. So this is the plant which shows in this is the picture which shows the inflorescence in cannabis. So this is the cannabis plant, and it is an inflorescence in cannabis. Next important function is leaf apenacity. It occurs when the upper surface of leaves grows faster than that of the lower surface. So जो upper surface है इसकी growth faster होती है as compared to the lower surface, then the leaf shows apenacity. Api means upon, nasty means moment. So the upper leaf surface grows faster than that of lower leaf surface. So when the upper side, that is the adaxial side of the petiole of the leaf, grows faster than the lower side of that is abaxial side, then the leaf curves downward, and this is known as leaf apenacity. So to lower side में curving है leaf की, this is known as leaf apenacity. This is called apenacity, and ethylene causes leaf apenacity in many plants like tomato and other dicot plants such as potato, pea, and sunflower. So this gaseous hormone is meant to induce leaf apenacity in these plants. And the young leaves are more sensitive as compared to the older ones. जो young leaves हैं वो इस hormones के लिए ज़्यादा sensitive है as compared to the older. Next is abscission of leaves, as we have discussed earlier during the classification of phytohormones. So ethylene and abscisic acids are growth inhibitory hormones. So it increases or it accelerates the rate of abscission. So it helps in the formation of this abscission zone. Ethylene promotes abscission of leaves in the plants, and older leaves are more sensitive than the younger ones because in younger leaves there is higher concentration of auxin, and auxin prevents the formation of this abscission zone. So the relative concentration of auxin on the two sides of the abscission layer has regulatory influence. It means when the concentration of auxin is higher on the both side of this zone, then this zone is not formed. But when there is decrease in the auxin concentration, then it accelerates the formation of this zone, that is the abscission zone. And that's why the auxin has regulatory influence on the production of ethylene that stimulates the leaf abscission. When the concentration of auxin is lower, जब ऑक्सीजन कंसंट्रेशन कम हो जाती है तो इथिलीन की जो कंसंट्रेशन दैट एनहांसेस इंक्रीज हो जाएगी एंड दैट प्रमोट द फॉर्मेशन ऑफ दिस एब्सिशन जोन. एट द टाइम ऑफ एब्सिशन जो कंसंट्रेशन है ऑक्सीजन की ऑन द लीफ लैमिना, जो लीफ लैमिना है उसमें ये डिक्रीज हो जाती है, एंड ड्यू टू डिक्रीज इन ऑक्सीजन कंसंट्रेशन इथाइलिन कंसंट्रेशन इंक्रीजेज एंड दैट प्रमोट्स द formation of this abscission zone, and finally it leads to the abscission of leaf. The important physiological response of ethylene is triple response. It occurs in case of some ethylated seedlings, and ethylated seedlings are those seedlings which are grown in dark conditions. So the triple response means three responses which include: the first one is shortening of the stem, that is stunted growth, second is the radial growth, and third one is the horizontal growth of the seedling that we will see in this picture, next upcoming picture. So this is the picture which shows the normal growth of the seedling, but when we apply ethylene in various concentrations, then we can observe these three responses. First one is reduction in the length of seedling, second is the radial growth, that is the swelling of the stem, and third one is the growth in horizontal direction of the stem. So these are the three responses which are shown by the treatment of this ethylene or due to ethylene. So ethylene causes triple response in the ethylated seedlings such as pea seedlings, which consist of the first one, inhibition of stem elongation, stem की growth नहीं हो पाती है अच्छे से, then stimulation of radial swelling of the stem, and the third one is horizontal growth of the stem with respect to gravity, and this is known as diageotropism. So this term is used for this movement with respect to gravity, that is diageotropism. So triple response affects the ethylated seedlings, and this was first to be related to the beginning of discovery of ethylene. So this was the first experiment in search of this hormone, that is ethylene, as a natural plant growth regulator, which is responsible for these various responses. Next physiological response of ethylene is plummular hook formation. So this is the picture which shows the plummular hook formation. The advantage of this hook is while penetration through the soil, this hook formation prevents the direct exposure of this apical region to the soil surface, which prevent the injury to this apical region. So the ethylated seedlings, in case of dicot plants, the plummular tip, that is shoot apex, is usually bent like a hook, and this is due to the presence of ethylene. This hook shape is advantageous to the seedling for the penetration to the soil and protect the tender, that is delicate or soft apical growing part of the plant from injury. So ये injury से directly बचाता है इसको, this hook formation. So this is advantageous during the growth of the seedling, and it is due to the presence of ethylene in the seedling. Next important physiological response of ethylene is formation of root hairs or formation of adventitious roots. This is the picture of seedling which is controlled one, that is इसमें कोई भी treatment नहीं दी गई है ethylene की. When we treat this seedling with ethylene, then we can see there is an enhancement or increase in the number of root hairs or formation of these root hairs in the seedling. So ethylene induces formation of adventitious roots in various plant parts. In many plants like Arabidopsis, which is a model plant, so ethylene treatment promotes the initiation of root hairs that we can see in this picture. So it's very important role of this ethylene, and it helps in formation of root hairs. So this is all about the various physiological responses of ethylene. Next, we will discuss about abscisic acid. Next, we will start with the abscisic acid, which is commonly known as the stress hormone, and it is produced under the stress conditions. This hormone is now commonly known as stress hormone and produced under the adverse environmental conditions. Stress hormone, and its production is stimulated by some environmental factors like drought, waterlogging, and other adverse environmental conditions to save the plants from adverse environmental conditions. It was formerly known as Abscission 2 because it is thought that it plays a very important role in abscission of plant parts like fruit, leaves, and other parts. So that's why it is formerly known as Abscission 2. It is also known as dormin because it is thought to induce dormancy in birds and some seeds and underground stem. So that's why this is also known as dormin. And it is detected in all the major living tissues of plants like root, stem, bud, leaves, fruits, seeds, and also in the phloem and xylem sap. So it is reported in all the plant parts. जो plant के सभी parts हैं उनमें इसको observe किया गया है. And it occurs frequently in the mature green leaves. जो mature green leaves है उसमें बहुत ज़्यादा होता है. And after its higher concentration, when it reaches its maximum concentration, then the leaf will shed due to abscission. Chemically, this is the structure of this abscisic acid, and it is basically a sesquiterpene, which means it contains 15 carbons, and it is a derivative of terpene, and basically it is a sesquiterpene. If we talk about its discovery, it was first observed by or isolated by Adicott in 1963 from the cotton balls. He observed that the cotton balls were falling down from the parent plant, and this chemical substance which was responsible for this abscission of cotton ball was abscisic acid. So in this picture we can see here the dropping of these cotton balls from this parent plant is due to this plant hormone, that is abscisic acid. So it is produced in many plant parts, but more abundantly inside the chloroplast of the green cells. Mostly ये chloroplast जो green plant cells हैं उनके अंदर ये ज़्यादा frequently मिलता है. And the hormone is formed by mevalonic acid pathway or from the xanthophyll. So this is the pathway from which this hormone is found in the plants. Next, some physiological roles of abscisic acid or function of abscisic acid. So first one is stomatal regulation. Abscisic acid control the stomatal opening and closure. Under stress conditions, the guard cells of stomata becomes flaccid and stomata are closed. So ABA causes stomatal closure under the water stress or drought stress conditions. It alters the osmotic potential of stomatal guard cells, causing them to shrink and results in the stomatal closure. So the basic principle behind this abscisic acid causes efflux of potassium ions. जो potassium ions हैं, they comes out of the guard cells, and it is followed by some anions and sucrose, and then there is a parallel conversion of some organic acids into the inactive form, that is starch, which is water insoluble form. So all these processes causes the exosmosis from guard cells. जो guard cells हैं उनसे उनकी exosmosis हो जाएगी, means जो water है that will comes out of the guard cells and goes to the surrounding cell, that results in the formation of flaccid guard cells, and finally the stomata becomes closed. So this picture shows here in this case when the stomata are open, the cells or guard cells are turgid. But under stress condition, this ABA causes the efflux, that is outward movement of these potassium ions. Then there are some sugars like sucrose and other anions that will comes out of the guard cells, that reduces the solute concentration inside the guard cells, and it is followed by the removal of water. That results in the formation of flaccid cells, flaccid guard cells, and the stomata becomes closed. So reduces the rate of transpiration. So it helps in controlling the rate of transpiration or water loss, and under stress condition, water stress condition, it controls the rate of transpiration. Next, seed dormancy. As we have already discussed, seed dormancy means when the seed fails to germinate even after providing the suitable conditions for germination. So it is controlled by the ratio of GA, that is gibberellic acid, and abscisic acid. It is mainly caused by abscisic acid, seed dormancy, and dormancy allows seed to tolerate desiccation and extreme temperatures in the better way. So it is advantageous process for the seeds. Under tough conditions, this abscisic acid helps in tolerating the extreme environmental conditions. So the buds as well as the seeds sprouts only when the abscisic acid is overcome by gibberellins. So ratio of this abscisic acid and gibberellins will decide that the bud or seed will germinate or sprout out. So it is controlled by the concentration of GA and abscisic acid. So this is the seed, which is a dormant seed, and when the concentration of GA is high, then it will germinate. If the concentration of ABA is high, then it will stay dormant. So the balance between ABA and GA is regulated by environmental conditions. If the environmental conditions are tough, then the concentration of ABA is high. Agar environmental conditions optimum hai, then the GA concentration will increase and it will stimulate the seed to germinate. So GA and ABA ratio controls the seed dormancy. Because of its action in inducing dormancy, abscisic acid or ABA is also known as dormin, that we have already discussed. Next is abscission, that is the falling down of leaf or any other plant parts from the parent plant. So it induces, this ABA induces the formation of this layer, that is known as abscission layer or abscission zone under stress condition. So it is known to produce this abscission layers at the base of leaf petiole where the dead cells are formed, and after that it will detach from the parent plant. So ABA concentration increases in the older leaves once the photosynthetic activity is halted or stopped. इसकी concentration ज़्यादा होती है in the older leaves, and abscisic acid promotes the abscission of flowers as well as fruits from the parent plants. Next is seed development. So ABA promotes the synthesis of storage proteins and lipids, and ABA content is very low, and during early embryogenesis, when seeds start germinating, during the formation of early embryo, so the concentration of ABA is very low, and it reaches maximum at the mid of embryogenesis, and then gradually falls to the low levels at the time of seed maturity. So in this way, the concentration of oxygen changes in the embryo of seed. So the seed germination is controlled by this ABA. When the conditions becomes optimum, then the concentration of ABA gets reduced and GA stimulates the seed to germinate. And this type of germination, when cotyledon comes out of the ground surface, this is known as apigeal germination. ABA has many other physiological roles, like it prevents precocious germination and vivipary. Precocious germination means when the plants start the seed starts germinating inside the fruit. So that is known as precocious germination or vivipary. It occurs in case of many fruits like tomato, in case of papaya and maize. So this is the fruit of tomato in which the precocious germination occurs. The seeds start germinating inside the fruit. And ABA promotes the root growth and inhibits the shoot growth in some plants. And deciduous trees undergo the phase of dormancy during winters, which is removed during the spring season. And this helps to overcome the adverse weather condition in the winters. So in winters, the concentration of ABA goes higher and it reduces in spring. So ABA level goes up during the winter season and the level of other hormone gets reduced. So AB basically helps in tolerating the tough environmental conditions. So that's why this is known as the stress hormone. So this was all about the roles of cytokinins, ethylene and abscisic acid. Hope you will get some idea from this presentation. If you have any questions, queries and any suggestion, you can give it in comment section, and you can also share it with your friends who need these lectures. Thank you. Have a nice day.